All right, we're going to get started again. Welcome to the new session. If we can get the first slide, there we go. Okay, I'm going to kick off and talk about how we can use AI in precision medicine and drug discovery. So let me start off with a problem that I've been facing for more than 15 years, which is the valley of death, we call it. So this valley of death is different than the startup valley of death. It's the valley of death for clinical drug development. As you all know, there's a lot of really terrific discoveries in biomedicine. They usually happen at universities and biotechnology companies. Somebody beavers away for a decade and they develop a small molecule or biological compound or diagnostic something, and they want to move that forward through the FDA process to get it to the clinic. That's that little bridge over here. Over on the other side is the market. It's the bench side. It's the value proposition. It's the saving lives. All scientists, all researchers strive to get their inventions, their discoveries over to that other side of the bridge where it's going to do some good for patients. The sad truth is very few inventions get across that bridge. Very little knowledge, very few products. Why? It's not the FDA's fault. It's not the regulatory environment. Although it is onerous, it is very difficult to go through the regulatory process and prove that your invention actually will help patients, and it's safe, and it's effective. That is difficult. But there's another problem, and the problem is heterogeneity of disease. Trying to position your drug to the right patient population, showing that it'll have effect in just the right population. And it was brought up earlier that one of the challenges that we have is there's no such thing as this representative population. We have to get much more intelligent about how to position drugs. So I want to show you one very specific use case of how you can use AI to accelerate this process. And it comes from Systems Imagination. This is a company that I'm associated with. I helped co-found with Chris Yu, who's in the back there, and he gave me permission to talk about one particular client use case, which is finding drugs to in-license. I don't know if I can go back here. So over here, there are more than 7,000 cancer drugs in development. 7,000. They all look the same. They have anti-cancer properties on cells. They cure cancer in mice. But how do you know it's going to work in the patient over there? If there was a way that you can find out very early, using big data and analytics and machine learning, which of those compounds, which of the 7,000, which one of the 7,000 you should invest in, that's important because this investment, going from this side to that side, that investment costs roughly $1 billion and takes roughly a decade. So it's not a trivial investment. You really want to make sure you choose the right compounds. So with Systems Imagination, the client said, let's mine the data out there, look at all the compounds, but let's take advantage of all of the public domain genomic data, clinical data, phenotypes, and let's find those few compounds that are worth advancing. So I won't go into the details of this, and we could talk offline if anybody's interested, but suffice it to say that once you take all the genomic features and extract the features, you can use all sorts of models to predict which targets are the best targets. So you could take all the cancer genomes and say, when we get drug resistance, which targets are highly expressed, amplified, and lethal? Those are my favorite targets. Highly expressed, amplified, lethal. That's a model. You can put that into the algorithms. But you need to organize the data to begin with. So let me take you through some of these approaches for managing data and collecting data. So you start with patient data, radiological images, bring in partners like imaging endpoints. You bring in partners like 3D scan on the pathology side, protein data, DNA data, RNA data. All of these disparate types of data need to be processed. They need to be translated from raw images and whole genomes into something that's useful. And that's what each of these swim lanes, each of these lanes represents a different software analysis pipeline that turns raw data into features. Because ultimately, a lot of these machine learning, a lot of these analytical systems, they require to begin with features. So once you have all of those turned into useful features, you want to integrate them into knowledge bases. So we have an integrated clinical phenotype database, integrated molecular phenotype database, but you also want to bring in prior knowledge. So we talked about TCGA, we talked about pathways, ontologies, literally any type of data that's out there that tells you something about what that gene is, what the protein does, how it interacts with other proteins is considered prior knowledge. So you take all that prior knowledge, all of that patient data, put it together, and then apply machine learning. The end result is you've taken all of this prior data, you computationally crunch it, and the question you're probably asking yourself is, did it work? Did we help the client identify compounds that would be prioritized? And the answer is absolutely. And the punchline in this story is that we found that something very unique in the data. We discovered that there is a whole theme, a whole set of targets that are vulnerable, and they have nothing to do with cancer cells. They're outside of cancer in the microenvironment. Now, Jeremy showed that if you use pathology and deep learning, you can identify these patterns. Those patterns often have something to do with the microenvironment. These drugs could potentially target cells that help, think about it as the soil. The cancer is the seed, you're targeting the soil. So we were able to find not only targets, but small molecules to in-license and help the client identify what they wanted using machine learning. This is just the beginning. From the other presentations, I think you're going to hear how we're at the stage now where we have a lot of genomic data, we have a lot of clinical data. The question is, how do you apply AI to deliver both personalized medicine, faster discovery of agents, and so forth? So we're very excited about this session. Let's start with David. David.
Can everybody hear me? All right, so my name is David Ledbetter. I'm a data scientist at Children's Hospital Los Angeles, and I'm going to talk to you today about attempting to leverage that data that we have access to in order to augment critical care medicine. So real quick, I'm going to talk about what's our overarching goal, kind of the philosophy we take with us when we approach that, the data we have to look at, the technologies we use. I believe I saw a question up on the board earlier about what is an RNN, so we're going to touch about that, our recurrent neural networks, and then a quick summary. So many of you might be familiar with this here. This is kind of the environment we're living in. This is what an ICU looks like when you go in. As you can see, lots and lots of monitors, wires, streams, lots of data flying at the clinician. Very easy to overlook something. In fact, you might have actually overlooked the patient that's actually right there in the middle. And so what we want to do is try and work on how can we leverage all of this data that's coming to us so that we can recommend the most effective treatment for a given patient. So when we do this, we do something that's kind of sacrilegious, and we actually have the clinicians and the data scientists working together. It's absolute madness. However, I think it's critically important to the problem we're facing. As a data scientist, we actually take the time out of our day to go and attend rounds frequently because it's really, really important for us to view and understand what actually goes on in the unit right now. That way, we can help the best way possible. Understanding what they do, how they acquire data, the amount of information that's exchanged between nurses and clinicians and parents is absolutely astonishing to the layman. And what is their decision-making process? What are their stumbling blocks? When do they go, I don't know if I should do A or B, help me provide some sort of insight into which path is going to be better. And then we also bring the clinicians in to consult when we're doing our actual algorithm development. They have all of this experience in the trenches that they can help bring to us so we can help understand, what are the clinically relevant problems we can tackle. Once we, come up with a conclusion, how can we actually implement that, and how we're going to make it so that the clinician or the nurse can actually use it and implement this into the workflow. So the data set we tend to work on mostly is EHR de-identified. That's been collected over about a decade. We have 20,000 ICU encounters that we look at. We have static information, things like demographics, encounter information, disposition. But the meat and potatoes of our data is actually this irregularly sampled time series, vital measurements, interventions, drug administration records that we use in order to make our decisions. And the technology we're using to make those decisions are these recurrent neural networks. So I only have time to give a fairly high-level overview of these recurrent neural networks, but it's effectively just a feedback loop. And what I mean by that is, at any time step with an arbitrary interval, we get all of this input, all of this data that's streaming to us from the ICU, and then we just generate an output. It might be, what do we think the heart rate's going to be at the next time step? Do we think this patient's going to survive? Do we believe this patient's going to be septic? And we generate an output, and then we also feed that output back in to the next time step, and we let this system propagate forward as a dynamical network. It gives us a lot of advantages in the ICU, really nice native comprehension of that temporal dimension. We do get the ability to operate on these irregular time grids because the data coming to us in the ICU, you never really know exactly when a nurse is going to capture the information coming to you. And it also has this nice dynamic temporal memory. You're not windowing yourself to what happened in the past 6 hours, 12 hours. You kind of get to maintain that history for the entire encounter of the patient. And we have these nice formal feedback mechanisms that is actually, if you remember Randall's slides earlier when we were making those predictions about which treatments do we think are going to be most beneficial for the patients, that feedback mechanism is the mechanism we're actually leveraging for that. And then it's very flexible. What inputs are available to us, and then what outputs do we want to go for. In particular, for today, I'm just going to talk about a fairly straightforward one, which is mortality. So that for our inputs, we can use that irregularly sampled time series data available to us, those vitals, interventions. And then for our output, we actually have a non-subjective truth, which is, did that patient actually survive their ICU encounter? And then in order to assess this, we can actually compare with the clinical baselines that they currently use in the unit, in particular PIM2 and PRISM3, for those of you aware of those baselines. So here we just have an analysis of the 12-hour mortality prediction. That is to say, once we've observed the patient for 12 hours, how well are we able to predict whether or not that patient will survive? We use that for convenience because it's kind of a baseline that's used frequently in the literature. We had 12,000 patients to train on. We actually had a test set of over 3,000 patients. And we saw really significant performance improvements over those clinical baselines, as well as a couple of more simple machine learning algorithms, logistic regression and multi-layer perceptrons. We use the 12-hour mortality, but you can also think about this in, as I see more and more information and history about this patient, how can I improve my understanding? And so here we just have the RMN prediction, and you can see even after only viewing the patient for an hour, you're coming near to those current clinical baseline capabilities, but as we increase from 3, 6, 9, 12, you're increasing your understanding of that patient through time. So just a really quick summary, we do have enough data and enough technology to leverage and make really interesting insights in the data. Using these, we have really significant improvements in our understanding of the patients as they go through our encounter. And the current work we're doing is using these models in order to generate understanding of both physiological responses as well as dispositional responses to the inputs and the treatments that can be given to these patients moving forward. So thank you very much.
Okay. I'm Giannis Ioannidis. I'm a computer scientist, an academic, and I'll say a few things about how we can help precision medicine or personalized medicine, as it was mentioned in the morning, and I agree with that, and drug discovery. I'll repeat one of my slides from yesterday, one of the workshops. We talk a lot about big data and analyzing data and deep learning and so on, but it's all not just algorithmic. It has to be part of a flow that involves a whole bunch of steps. First of all, the data that goes in, the information that goes into any of this process can be bottom-up, so data that you collect from the hospital or from any other source, but also top-down. There's a lot of medical knowledge in the heads of physicians or in ontologies or in other sources, and both of these have to be taken into account, not just the data that you collect. And if you look at the steps, okay, raw data, the bottom-up data, first of all, you have to pre-process it. I saw the term curator in that wonderful curation, in that wonderful graph that Spiro showed. Well, you have to curate, you have to clean your data, you have to massage it and prepare it for what is going to come next. And then knowledge discovery, data mining, machine learning, and so on may come in to just grab the trends that you see in the data. And that then comes to modeling. And for that modeling, you need a lot of domain knowledge that has to come from above. And you can model the heart, the various pieces, the tissues, and so on. That's the dream that will reach that there, so that together with the data and the models, we can come back and finally reach the personalized model-guided medicine, where you can reason and say, this particular case, how similar it is with another case, or the 150 patients that have the particular lung, and so on. Unless we implement such a workflow, or this one or someone else, we are working on this particular one, then a lot of information will be missing. Let me focus, by the way, all this is, obviously, what you observe in the end has to come back and feed into the raw data or the domain knowledge, so that you can improve your models, you can improve your algorithms, and so on. One of the domain knowledges that exists is publications. A lot of the medical knowledge has been expressed, has been recorded into publications. What we can do with that? We've been working on analyzing PubMed, or whatever is available in open access, or we can get our hands on, together with other domain knowledge that are in semantic bioterms, protein data bank codes, mesh terms, and so on and so forth, and taking all this, analyzing the text in these publications, using the other pieces of information, mining the graph that comes out of connections of all this information, and also doing modeling of the topic of each paper. So not just analyzing the language and the semantics behind it, but trying to understand what kind of topic is each paper on, and using that as the conduit to come up with a graph of all the possible entities that exist expressed in these papers, and from all that get to some interesting results. So let me just give you briefly what is multi-view topic modeling. Usually, when you analyze and text mine papers, you just look at the terms, and you identify, what is in the text. But if you come and say, what is this topic about? You can only reach up to a certain level of sophistication if you just stick to the terms. But if you bring in a whole bunch of other semantic information, like particular phrases that are important to your field, or mesh descriptors, or the journals where the paper is published, then you can come up with a topic that is described by elements from all of these dimensions, and then a human, and that goes the curation, can come up and bring in an intelligible expression of the topic like this. And this is another case where you bring in also PDB codes and mesh descriptions, what your dimensions is, what the kind of entities that you can bring in to help on top of the text is up to you. And now this whole process, how it works, you analyze and you do a whole bunch of things, or you model your data by looking at the text, identifying entities, seeing connections, seeing particular entities in the categories that you want. And then you identify a topic. And as I said, the topic is the conduit for connecting all these and creating a graph where you find similar entities, clusters of entities like this, and the nodes in the graph, again, is a matter of your choice, what you are interested in connecting. It can be drugs, protein data bank, proteins, mesh terms, you name it, patients, regions of the world, if you are into epidemiology. These are the nodes, and you connect them based on the similarities of the topics of the papers that you find them in. And then you can categorize them and all that. And through this, through the graph, you can find potential drugs that could help. Let me give you an example. What I described before is something that we are involved in with a bunch of European partners. A similar effort is done by Indiana and Tsinghua, and you can see through these topics of the papers, you can see drugs mentioned in the papers, genes mentioned in the papers, and various diseases. And you can find solid lines that have been identified and exist as well known in the literature that, for example, this gene is related to this drug and is related to this or the other disease. And you can see potentials. The analysis of the publications show you that there is a potential relationship of this and this. Do we believe that? No. We have to go back and then test it in the traditional way, but this provides you targets like Spiro was talking about. And this is another example. Last example from IBM and a bunch of others. A particular protein suppressor, P53, a bunch of kinases were known that they do phosphorylate that. These are the green ones. But from the literature, a bunch of yellow and orange ones seem to be related. Again, targets for this. And going back to the lab, I'm wrapping up, that's the last one. Going back to the lab, it shows that what has been identified in the literature indeed helps and does that. From this, we go back to the loop. You find something there, you go and do clinical trials, and you have similarity of patients. Thank you.
Okay, so I'm Mei Wang from biomedical engineer of Georgia Tech and Emory University. So I want to actually start with, when we're talking about precision medicine, what exactly the word came from. It's actually said delivering the right treatment at the right time, every time, to the right person. The Precision Medicine Initiative will leverage advances in genomics, emerging method for managing and analyzing large data set. So basically, the US government invests $250 million on this. This is what we foresee, what's happening. Yesterday, the current medicine is, you use it, we call it intuition medicine. You use symptom-based to do diagnosis. Now, we're talking about the pattern-based, evidence-based medicine. In the future, we're talking about algorithm-based precision medicine. In this whole process, we're actually talking about how to use a personal molecular fingerprint plus objective quantification plus the data analytics, including both data mining and AI, to help us get to precision medicine. With that, yesterday on the workshop, I actually gave an example on precision medicine for using pathological imaging diagnosis. Today, I'm going to focus on the genomics. So this is the survey in the community right now. We're talking about clinical personal genomics or clinical genomics. The number one challenge is the entire field is facing. There are so many tools and methods. Nobody knows where to start, even when we're talking about precision medicine to use the genomic information, right? So that's the reason why the major organization that's facing this is being mentioned multiple times today. That's called reproducibility and the regulatory process. Because FDA is the one facing the big challenge. They need to regulate medical devices, but how do they regulate AI algorithms? So that's actually the reason why they formulated this international consortium with 36 teams around the world, and we actually started investigating what's going on. So let's take a look at what's exactly the challenges. You're talking about using RNA-seq as an example. You're using the samples, you have different platform, you have lab technology, Roche, all Illumina, and then you have so many algorithms, right? And then totally together, combining all the potential like algorithm platform, how many possibilities are we talking about? Making a guess. Actually, 72,000 possibilities, right? So the FDA wants to know how do they regulate? What's the data analysis algorithm they should use to regulate the medical genomics? Isn't this sounds familiar? Isn't this also one of the biggest challenge? Which is, how do we find the decision actionable algorithms? So quickly go through this. As I said, this is actually unpublished work because this is a state-of-art conference. That's why I'm sharing the result as a result of this whole consortium. Out of 72,000 possibilities, we actually extracted 278 representative algorithms. Together, the fundamental key message about our AI or machine learning for precision medicine is ultimately go back to a simple thing. That's called performance. It's all about trade-off. How do we trade off different possibilities? Of course, the first thing we have to do is, coming from the medical genomic, has to go back to medicine. So we have to follow the gold standard. So accuracy is one metric. Second one is called precision. How do we actually know reproducibility can be guaranteed in the medical genomic process? Third one, sensitivity. How do we detect the low expressed genes compared to the regular expressed genes? Last one is specificity. How do we do the false detection rate for differential expressed genes? For the next 3 slides, I'm actually going to just quickly show you what exactly being selected rather than diving into detail. So for example, you're talking about a aligner, how you map the 3 billion base pair to reads. You're talking about some representative mapping tool for aligning. And if you're talking about quantification, you also have multiple choices. And together with the normalization, typical normalization, totally together, that's when we get the 278 representative one, focusing on the reproducibility, repeatability, and the biological utility. Because, as mentioned in the speaker this morning, every single algorithm, machine learning, is data structure dependent and the clinical utility dependent. We actually need to work on that based on the trade-off, as I mentioned earlier on. And of course, ultimately, it has to be clinical actionable, so we have to go through the clinical knowledge. Okay. Quickly show you, as I said, this paper is under review. I'm just going to quickly point out how critical it is to understand. I think it's Randall mentioned about the black box, should the clinician know it or not. My recommendation, you better know some, if you're not all, because a simple example is, with using just one algorithm change in the aligner result, your clinical, originally like a separable, like clinical cases can no longer be separable. By the way, this work is being under review under Nature method. We're actually going to share this multi-year investigation on this. One more thing is, Randall mentioned early on about guideline. When you have a black box, you need a guideline. At least right now, FDA wants to regulate, but right now it's very hard to develop something like the medical devices. So that's the reason why we want to provide AI methodology guideline so that we can have the clinical reproducible results. Okay, so now I'm actually wrapping up my talk, but I do want to mention the last mile issue that's also being mentioned early on, which is after we do all the investigation. For example, for this investigation, we put 200 million CPU hours, created hundreds of terabytes of data for 278 next-generation sequencing results. Of course, right now, we need to, I said I didn't have time to cover the imaging, but we actually, in the real clinical validation, we have to do that, combining with the pathology diagnosis to validate it. Eventually, the key thing is delivered to the clinical side, so it can be utilized in the clinical setting. For that particular reason, I'm actually highlighting an important effort. It's called an emerging effort, called FAST, like Health Informatics Resource, FHIR resources. This is a new HL7 standard. The key thing is how do we bring the genomic into the EHR besides and beyond make all the existing EHR compatible. So with that, I think I covered this basic message. This now, in the whole country, USC, like Dr. Francis Richmond, he lead like a regulatory science discipline. Other than that, we haven't been able to have much regulatory science. But I think for AI to be medicine regulatory has to be a top priority. Thank you.
That's great. Thank you to all the speakers. Well, we're going to take some questions now. Let's start with the top one for Yannis, I assume. In the publication network analysis, was there a semantic component to the natural language processing, and how important is that?
It's all about semantics. I'm not sure what a semantic component would be, but certainly bringing in ontologies or taxonomies of terms is very important to connect, independent of the particular term that someone is using, to connect to the overall concept and then understand what it is about. Without semantics, you can achieve, but not as much. It was definitely necessary.
Okay. Now, The second one talks about traditional researchers criticizing real-world data as having poor quality. Are there thresholds where the volume makes up for the poor veracity? Who wants to take that one?
Since I hold the mic, let me just say, I don't think there may be an absolute threshold, but I think that certainly the more data you have, the more clean data you have, you can afford to have wrong data as well. But there will always be cases that even one wrong data point may screw the whole statistics and may give you wrong results. So I cannot imagine having absolute thresholds that will help you decide, yes, I'm fine or not.
I think it would probably be one problem is specific, but the other component is always going to be cost. Sometimes just collecting a large amount of data is extraordinarily cheap, and so you can afford to just throw data at it, where at the same time actually truthing any of that data can be extraordinarily expensive. And so if you need to get a radiologist time to truth your image, or if you need a team of radiologists to give you a community opinion on the truth of a particular data point, that can get very expensive very, very quickly. I know for our own work, we think about kind of diagnostics. However, most of diagnostic codes are captured at the encounter level. Ideally, we'd like temporal diagnostic information, but when we do a back-of-the-envelope calculation about what are the costs in order to get a team of physicians to sit down case by case and put temporal markings on diagnostic information, it just becomes infeasible very, very quickly.
So maybe I can also quickly comment on this question. So in fact, I would answer that there are no thresholds where volume can make up for the poor veracity. The reason is simple. If you look at the literature in the electronic medical record data quality issue, and then the early models was talking about you actually doing the data imputation, trying to deal with the noisy and missing data. The reality, however, most of the model failed over the last 10 years. And the real reason is because all the data quality have different property coming from different reasons. Some of the data quality are coming from the secondary derivable data. For that kind of thing, you have large volume, it's going to make up. Some of the data, once it's missing, is actually fundamentally important to the particular patient group. There's no way you can make up by using other patient group to do that. So that's why it has nothing to do with volume. It has something to do with the category of the quality issues.
That's great. A little bit of a poetic question for David, I guess. To impute or not to impute?
That is the question, isn't it? I think it actually goes back to May's point that she brought up in her presentation, which is it's really important to make sure that you have a proper quantitative framework. If you put things together in a modular fashion, it should not be difficult to have your imputation step as a module that you can just plug and play. For instance, we did a lot of experiments with our particular framework, some based off clinical guidance. One thing that we were told was if we're not taking a measurement, it's because we believe it's stable, such that a simple forward fill imputation is frequently the proper approach to take. We didn't necessarily take that as gospel. However, we took that under consideration, and all of a sudden, as long as we have a proper quantitative assessment framework, we can try a forward fill imputation. We can try a linear imputation. We can try, a Gaussian method in order to pull out particular data points. And then the assessment is really where you get your answer. Did we actually perform better on new patients in order to, learn something about them moving forward? And I find it very difficult as a data scientist in order to ever attempt to make that judgment a priori. I always want to take a measurement, be as objective as possible and say, well, the data says this. You might look back and say, well, there's a bias in the way the data was collected, which led you to that, which might not hold in another institution. But then as a data scientist, my answer is, well, get the data from another institution and we'll rerun the experiment.
One of the earlier questions was, for drug discovery, is AI better at picking winning compounds or identifying loser compounds more quickly? That's a very interesting question. It seems logical that you could use AI in both directions, but pragmatically, you don't need AI to pick losing compounds. Most of them are losing compounds. What you do is you use very simple filters. You pick criteria, you use very simple algorithms to find compounds that have features that would flag them as not likely to succeed. What's much harder is finding those, forgot who mentioned it, the unknown unknowns, finding those insights into cancer biology that might not be recognized. So there may be drug targets that the chemists that developed the inhibitor to that target may not recognize that target as being involved in some biology. Finding that unknown biology, positioning drugs to the right indications, that's where multiscalar modeling and more complex analytics, AI, could be more useful. Looks like there's a question about top-down versus bottom-up. How much of our modeling and training will require expert top-down versus data-driven bottom-up methods now? When will we move to a primary data-driven model? May and Yanya, do you guys want to field that one?
I can quickly answer. And then.
Yeah.
So this is actually a very good question being discussed a lot. And I give you a simple example. You're talking about Boeing 737. You put all the component, electronic, mechanical elements. Can you actually build a plane? You cannot. You need actually top-down to design it. So that's why for this particular question, it's always about we need to really integrate. But exactly what's the process? So this is actually the process for biomedicine that actually works more efficiently, which is the first step. It needs to be top-down first. It has to be driven by the medical need and it has to be validated by medical knowledge. So with that, when we do mining, we use knowledge-guided and knowledge-driven AI model development for the first phase. And then after that, we can use actually AI model to do what we call unsupervised exploratory learning to discover more data insight because you already build a confidence in our user community who actually, can you prove this black box, the new models you're developing? And then the third level we always talk about in the data analytics, it's very important ultimately to develop a biological inspired AI model, such as neural network, all genetic algorithms. So that's why it should be a spiral process.
Yes, I agree. I don't think we'll ever go into purely data-driven model. You need the top-down and the bottom-up. Just a quick example. From a bottom-up approach, you cannot distinguish on whether cancer causes smoking or smoking causes cancer. It's a very simple top-down information that the causality goes in the direction that we all know. Expand that into more complicated cases, and the top-down knowledge, not just for causality but for other reasons, is crucial in order to make interpretable, much better interpretable, the information that you get in bottom-up. So we need both, and the trade-off, I mean, the intensity may be different in each case, but there will always be both.
In the workshop session yesterday, we actually talked about, we did a real experiment where we had a clinical top-down pediatric early warning system baseline, and we actually compared that to kind of our data-driven model, and we were actually able to quantify at exactly what point did we have enough data where the data-driven model actually outperformed. It's a really interesting experiment that I think can be performed on a number of different tasks, and if you're interested, ask me. However, I kind of have to segue this question into another question because I'm on the panel, so I get to ask questions if I want, right?
We're almost done.
It's not going to be enough time. Two contradictory things that we've heard a lot about today. One is p-hacking. The other one is the incorporation of subject matter expertise when we're going into building these models. And maybe it's a question beyond the scope of this particular panel in particular. But how do we bring those two things together when so much of that literature has kind of those p-hacking elements that make it difficult to integrate and really appreciate which ones are true positives and which ones are false positives that we're attempting to bring into our fold.
So I think that's a sunset question. So let's take that to the sunset. I want to thank the speakers for just a terrific session. And I'm just going, thank you. Just going to announce that we're all going to go outside and see the sunset if you like. It's really spectacular, really recommend it. And then please do come back right after the sunset. We have another session, so don't forget, don't get lost. Come right back. Thank you.